Wonderful, thanks so much. I'm a professor of radiation oncology and co-founder of Modifi Bio. Really excited to tell you about the story. So for this slide, it's just the high points. We're doing something that no one is doing in the oncology space right now, which is exploiting cancer-associated DNA repair defects by DNA modification. We have a strong team, and we have a lead asset that's headed into the clinic. We're candidate nomination imminent in the next 2 quarters. We hope to be in the clinic by the end of next year. We are not only a lead asset focused program, but we also have a platform technology that we can apply to many different DNA repair defects. And we're currently raising the seed 2 round, which we hope will support us to nominate that candidate and close our Series A later this year. In terms of the team, I'm one of the co-founders. We also have Seth Herzon from the Department of Chemistry at Yale, Kingston Lin from the medical school, and my longtime business partner, Kevin Rakin. Kevin and I have started a few companies you may be aware of. I'm one of the co-founders of Cybrexa Therapeutics as well as Alphina Therapeutics, and this is our next big thing. For the team, just want to show you those logos. What's really exciting for a seed stage company, we've been able to recruit people from publicly traded companies to come to New Haven to go work for us. And similarly, we have an advisory board on the SAB of KOLs crossing the bench to bedside space and the glioma and the non-glioma realms. So what exactly are we doing? So in the DNA repair synthetic lethality space, we'd argue that almost everyone is indirectly targeting proteins. They indirectly target those proteins to create complex DNA lesions that DNA repair defective cells cannot resolve. We're going back on that central dogma and actually modifying the DNA directly. So I'm in clinic today radiating patients, and we radiate the tumor DNA, and it works really well, but the therapeutic index is the problem. And so how is Modifi going to solve this? Well, we have a core technology which we call DNA modifiers. These are like ADCs or peptide drug conjugates, but very simple orally administered molecules consist of a vehicle and modifier. These release the modifiers, bind to the DNA, but we iteratively refine them so normal cells can rapidly remove them, but cancer cells with specific DNA repair defects are unable to resolve them and subsequently die. We have a lead program just at a high level today, MGMT, called methylguanine methyltransferase. This is a gene that's silenced in many different cancers. Temozolomide and conventional chemotherapies work really well in this space, but they require resistance via mismatch repair mutations. And the mechanisms on the right, what I'll tell you is if we can find a DNA modifier that can exploit the MGMT defect and be impervious to tumor-associated mutations, we'd have a therapeutic win. That's exactly what we found. So this has been an academic collaboration that moved into Modifi. We published this paper in Science just last summer. And we showed here, I'll just draw your eyes to the green and the yellow curves here. You can see the MGMT-dependent interaction impervious to mismatch repair mutation status. Very, very nice effect in vitro that we also see in vivo. So I'll show you here on the next slide, this is a PDX model with our genotype that's resistant to all the standard of care therapies. Intracranial xenograft model, 3 cycles, monotherapy efficacy, 8-fold improvement in overall survival. I've been working in the lab, running the lab for about 10 years. We haven't seen data like this in quite some time. We, in parallel as a clinician scientist, working with my SAB to develop our clinical trial, because we will be in the clinic in about a year. You can see here precision oncology basket trial design, which we're very excited about. And then we'll just end here with our drug development strategy overall. As I mentioned, what's unique about the seed stage company is we have a candidate nomination imminent, and that will put us in the clinic by the end of next year. We completed all this work with very financially disciplined strategy with a seed 1 round of $6.4 million and a phase 2 SBIR. We're now raising the seed 2, currently at $4 million, but we're extending it because of the investor interest. So certainly reach out to me if you're interested. And we hope by closing that seed 2, we'll nominate our candidate and be able to close our Series A by the end of the year. So I'll just leave you this as a visual. I'd say we're very excited about an entirely new way to target DNA repair defects in cancer based on high impact cutting edge science at Yale. And we're very close to the clinic, and we're excited to extend the platform out to many, many different DNA repair defects. And so definitely reach out to me if there's any interest. Thank you.